Okay, we'll go ahead and get started. So first, let me say thank you all for coming. I want to say for folks who are currently in the faculty market this year, I want to recognize that this is a tough year, but also that we're seeing just kind of disruptions to the cycle, that things aren't necessarily going on the timeline that we normally see, and that we're seeing positions open that are delayed from where they normally would be opening in September or October. And so what that means is if you aren't seeing a lot yet, I don't think that means that it's time to start planning for next cycle yet. I think keep looking because we are seeing positions come out that things are just seem to be pushed back a little bit this year. Also, if you were in the faculty job search group with us since July, thanks for being here. Thanks for sticking with it. And I know that there were a lot of kind of check-ins and meetings that were postponed because of the shutdown. Emily is working on rescheduling some check-ins for January. So we do want to hear from you all how it's going, and we'll get those scheduled soon. I am very happy that today we have Todd McFarland to talk about Chalk Talks. I know some of you may have interviews coming up. Some of you may still be hoping for interviews because the cycle has been so strange this time around. Um, but this is something that is really going to be helpful when you get to those interviews. So Todd is a senior investigator in NICHD, and he leads the section on mammalian development and evolution. And he has helped a lot of our fellows through this process by giving talks just like this, and also by helping with faculty packages. So Todd, I'm going to go ahead and hand it over to you.
Sure. And Laura, can I ask you a question? So,
Sure.
one of the things that we do in our division, in our group is, we have open pages where people post job opportunities all the time. So in other words, so you're not, just depending upon yourself searching. I guess that does OITE do this? Is there like a place where people post things they find on social media on?
Yeah, so we don't do that directly within the group. People certainly share, but what we do really recommend is using Future PI Slack, which is
Yes.
where you can find lots and lots of, postings, lots of advice, just see what other people's experiences are. Are people getting interviews yet? How's it going in the cycle? So if you're not on Future PI Slack, I really recommend that.
Yeah, 100% agree with that because you're getting like that real-time information about all things related to job searching. So, okay, good though. But let's get started on what I was what I was tasked with doing today, which is to talk to everybody about how to give a good chalk talk, or to sort of demystify the purpose of the chalk talk, and how you might prepare yourself to give an effective one. And this is a little workshop that we started just with my own institute's affinity groups, where we started with small numbers of postdocs who are, on the job market. And basically the what I put into the content of this is really based on my experience when I was on the job market about 13 years ago. And not a whole lot has changed. And I can say that because I've also served on Statman committees almost every year since I've been to NIH. And the NIH Statman searches almost always had like a major seminar where you have about an hour to present your research, and that's typically followed either right after, but usually the day after, where you'll give a chalk talk um, related to what work you're proposing to do if you get a faculty position. So I've seen dozens of chalk talks. I've seen ones that were all the way from spectacular chalk talks to, I won't say disasters, but nearing towards disasters. So what we're trying to do is to avoid that by helping you to really get prepared. And I think that's the key thing. Like anything you do in science, when you talk about presentations, is you should go very prepared to give your chalk talk. And there's a lot you can do to get ready. So this is also meant to be a little bit of a workshop, and I'm going to ask everyone, if you don't have it, to get a piece of paper, a pencil, something, because we're actually going to do a mini exercise in the middle of this to get you thinking about how to present a chalk talk. Okay, so I hope my slides advance. I only have 7 slides, so a lot of this is going to be discussion, so I hope you'll also ask questions at any point during the seminar. Okay, first, what is the purpose of a chalk talk? And I really break this down into 2 major items. The purpose is so that you can present your vision for your exciting and groundbreaking research program. So it's sort of like where am I going? And the second is really to convince the department or the institute that you are prepared to lead a research group. All right, so those are the two things that you're trying to achieve. For the first one, what you're really going to be doing in the Chalk Talk, though, is really outlining your short-term and your long-term goals. So in the short term, you're going to be basically describing the major aims in like your first R01 style grant. Now, normally when you've applied for a faculty position, you've already written up a proposal that probably looks like an R01 grant. And I'm sure Laura and and the OITE has offered some suggestions about how you're going to do grant writing. So I'm not going to go into all the details about R01 grant writing, but most of you should be familiar with this. The second thing, so that's the short term. The second thing you're going to be describing is what will be the impact that your group will have on the field. So you really have to think about the big picture. What are you going to be known for? What is your niche? What is unique about your group from those groups who may be your competitors? Now, the second thing you need to do is to convince the department that you're prepared to lead a research group. So you have to have thought about, hopefully, about who are the kinds of people you want to hire and why are you going to hire them? So are you planning to hire postdocs, postbacs? This basically shows that you've done your research about what is the primary type of people at that institute might hire. So if you're at like a smaller college, you might be working more with undergraduate researchers. But if you're going to go to like a big R01 medical institute, it's possible you might have graduate students, medical students, or postdocs in your laboratory. The second thing is you have to have really thought about what resources you will need to be successful. What do I mean by that? Well, if you're a laboratory that does confocal microscopy, then obviously you need to be able to describe like the kinds of equipment that you'll need. But it may be that you want to do clinical research, and it's important then that you have the right types of clinical support for your kinds of research. So this could be wide ranging, but you really have to think about what is the institute going to need to provide you with in order for you to be successful. And then you also want to think about how you're going to focus and divide your efforts amongst your group. So have you thought about which type of people might tackle which particular aims in your project? And these are things that you sort of want to be able to present while you're going. Okay. The second thing is, how do you prepare for your Chalk Talk? And I really think that one of the most important things you can actually do is, if you're fortunate enough to get asked to come to an interview, it's absolutely fine to ask a question to whoever invited you about whether there will be any specific things to the formatting of your Chalk Talk. So for example, are there PowerPoint slides allowed? Are they expected or maybe encouraged? Are you only going to be drawing? And I think it's important to know, like, where, if you can at least, what type of drawing? Is it going to be on a whiteboard? Maybe you should bring markers along. Maybe it's going to be a chalkboard. I think it's very reasonable to ask, what tools am I going to have? Because it's nice to know, to plan ahead about what's going to be available to you. It's also good to know how much time you'll have to prepare. Typically when you're giving your Chalk Talk, it's on the same day that you're going to have lots of one-on-one interviews. So, do you have 30 minutes ahead of your Chalk Talk to really put things out on the board? Or are you going to be rushed there and have maybe 5 minutes? So it's important to know if you're going to be doing most of your drawing on the fly, or if maybe you have some time to put some stuff on the board ahead of time. The other thing you want to know is who's going to be attending. And not that they're going to be able to tell you everybody in the audience, but in some cases, it may be just limited to a very small number of faculty. It could be a very open session. And it's really nice to know, basically, anytime you give a presentation, whether it's a seminar or a Chalk Talk, it's good to know who's in the audience so that you might be able to tailor it a bit better. Now, the other key thing is that you really have to think about and plan and practice how you're going to sketch out your aims for your R01 proposal. And one of the things that I really recommend doing, some people have done this in talks that they've given out, but you're certainly able to prepare a document that you bring with you. And I like to do this when I gave my Chalk Talks and my presentations. I had a very small paper that had a little bit of an outline so that I could hand it to everybody who showed up. And that way they had a piece of paper that they could take notes on. They could follow along with what the aims are. Now, you don't want to make that like a 10-page thing. I recommend doing it in 1 page, but it's just something for the audience to look at while they're following you along on the Chalk Talk. Okay, what about the content of the Chalk Talk? And I can't tell if anyone's raising their hands, but if anyone has a question, feel free to just turn your mic on and stop me, or you can turn on your video, and I'll be able to see you.
Yeah, I think, Todd, I don't think they have the ability to turn their mics on, but if you all
Oh,
do have questions, you can put them in the Q&A. It looks like this was set up as a webinar, but
oh,
I'm
okay. So
happy to
I
read out
can't.
from the Q&A.
Oh, bummer. I can't
Yeah,
listen.
I'll see if I can
Okay.
change it.
Okay. I miss Zoom. Um, okay. So the Chalk Talk content. So I keep mentioning this R01 style. What I really mean by that is, typically you're going to have prepared three aims. So I have a question. Hold on. I saw something pop up. I lost it. Okay. Um, so you're typically going to have three aims. And what you typically want to do is first you want to set up something about what is known in the field. What are the big questions that you will address? And these are really like the sort of your long-term goals. Then you want to consider what are the short-term goals that you have. And typically what you're going to do is you're going to list your three aims, and then you're going to attack them in order. And I found that in most cases, you want to list your most important aims first. So you want to tackle them sort of in the order of importance, because typically you'll have more time to talk about the things you start about. So for aim one, for example, it's going to be the thing you spend the most time on. Aim two, slightly less, and aim three, the less if you can get to it. Um, it's important that you're spacing your talk so that you'll both stay on time. So you have to know how long do I have? And then you'll also have time to discuss all three of your aims. Now, this is important when you're writing a grant, but it's also important when you're describing them, is that remember that your aims should be conceptually linked to each other. Obviously, they're all written in a similar grant, but each aim should not be dependent on the other's success. So you can't make an aim two that's dependent upon aim one being successful. It's important that you're stating your hypotheses clearly and that your experiments are designed to give you clear answers. So typically like a yes or no kind of answer. And finally, you should be prepared to discuss alternative approaches. Now, all of these things are probably not that surprising to you because most of you have probably written grants, and these are all things that you typically want to put into your grant. But you want to make sure that you're also getting these concepts across while you're presenting the Chalk Talk. Okay, now I want to take a few minutes now, and I want to ask everyone to grab their pencil or pen and a piece of paper. And I'm hoping that everybody can take a few minutes and give me some sketches and tell me what your Chalk Talk is going to be about. All right. So I've got my pen, and I'm going to do this exercise too. I haven't practiced this. I want everyone to think about what they're going to draw on the chalkboard for their Chalk Talk. All right, go for it. And I'm hoping that at the end, somebody could share their drawings for me on the screen so we can see some of the things that people have laid out. I'm going to shut off my video just while I'm drawing, but I'm still here. Okay. I'm going to show everybody my visual. I only took a minute or so. I'm sure some of you were still writing. See if everybody can see my paper. You guys see this? I know you can't read it probably, but I have a very simple schematic drawing on the bottom. It's got some genes with some promoters, a couple of proteins, and I have three relatively short questions, which represent the major questions of my three specific aims. So what I tried to do is to keep my questions pretty simple. So let me just read them to you so you guys can see this. You don't have to know that much about my project for this, but I wrote, how do crab zinc finger proteins regulate protocadherin gene expression? That was my aim 1. Aim 2, can crab ZFP binding site mutations mimic KZFP knockout phenotypes? And then third, can we manipulate barrier function by crab ZFP protein recruitment? So I had three sort of simple sentences that were all questions, and I had a relatively simple model. Okay. All right. Can somebody else in the audience who wants to be brave speak? I actually don't know. Laura, can we do this or no?
Yes, yeah, they should be able to now. So you all can unmute and hopefully turn on your camera as well.
Yes, I would love if somebody would volunteer. This worked well last time we did this. I hope somebody's brave enough to do this. I can say I was brave, but I knew the assignment before going in.
Yeah. Hi, Doug.
Great. Who's that?
Yeah. So I'm Ruchi, and yeah, currently I'm looking for a job, so I attended this. So I don't know if you can see my drawing.
Oh yeah, great. It's like a little diagram with arrows
Yeah.
and a few words. That's great
Yes.
because it's not super complicated. Good.
Okay. So
Yeah.
I write my the big question. So I work on the circadian rhythm. So
Okay.
wrote a big question like how the suprachiasmatic nucleus, which is the master pacemaker, regulate the brain-wide circadian output.
Yes,
Okay.
love it. Yeah.
And then I made a flow diagram. I put a retina that send the light information to the SCN, and then it connect to the different hypothalamus and then regulate like feeding behavior and metabolism. Okay, then I ask my question here, what is the gating that SCN receive the light input? And then what is the synaptic plasticity? Maybe I'm going too much
No,
in
that's terrific.
terminology,
I love
but
this because it's
yeah.
relatively simple still. So this is good.
And then I ask another question. These are my short term, like what is the glia neuron interaction that is
Yeah.
involved in the circadian rhythmicity? And then I ask a very broad question like how the SCN2 brain output. Like this is like broader question that I'm asking, like very big
Yeah.
question. Yeah.
Yeah, this is perfect because it really hits on the major questions that you're interested in.
Yeah.
And you can very easily see how after you start with that big picture, how you're going to explain how you start to address those and the methods that you're going to use. Yeah.
Mhm.
So I think it's very important that you sort of outline the big picture of the three aims, and then you can kind of tackle the individual components as you go. So I think that's a terrific start. And I can tell that the model was well thought out. It was relatively simple. It didn't have too many words, but you need to be able to write some things down. So great. That was great.
Thank you.
How about somebody else? Can I see another one?
Todd, can I make a suggestion?
Sure.
Can you try stop sharing your slides? I think
Oh,
we're
yeah.
going to be able to see
Yeah, even better.
the individuals better. Yeah.
Yeah, I can do that. How do I stop sharing? Let's see. You know how to stop sharing? Is there a button on the top? Oh, there it is. Okay, I got it. Yeah, great. Okay. The problem is that I don't know if anyone can share easily from the picture. Anybody else want to try? I might not get any others. Well, I'll tell you what we can do. I can keep moving. And then
I
I'll be
can
happy.
volunteer.
Oh, yeah, great. Who's this? Johan, is that you?
Yes, if I can
Okay.
get my camera going, just a
Yeah.
moment. Hi,
Great.
sorry. It's still very simplistic. I'm not
Yeah.
sure. Probably have to do the blurring of my camera off.
Hold it out in front of
Sorry
your
about-
face. Hold it in front of your face. Oh, that's good. I can see that.
Oh,
Ah,
that's
good.
it?
Simple. Yeah, simple. Good.
So the basic, okay, here. So the basic idea behind this is that I found that repeated exposures to Ixodes scapularis, which is a tick, causes a local itch that allows you to detect it and remove it very quickly. And building on that study, I want to know if other tick species can induce the same mechanism. The interesting part here is the saliva, which are the active molecules which induce this response, are very different between tick species. So that's the first question. How, if they are, doing all the same thing, how the immune response to all these different salivary proteins actually channels into the same neural response. So you can have your itch, and that needs to be mediated to the brain. And the brain then tells you to scratch. So that's a neuroimmunological response. So it's a relatively new field.
Yeah.
And the second question out of there is, of course, dissecting the immune response, whether the tick response or you get a response to a tick species or not. Both can give you valuable insights in terms of how the mechanism actually works. And the third question is if there is actual cross-reactivity. So if your aim is to develop a vaccine, you want to have as simple as possible vaccine and not a very complex one. And that basically are the 3 levels in the study. And it would always go through the same experimental cycle, so to speak.
Yes. So I like that too. That's very well thought out. It's higher level so that even a non-expert like myself in that field really understands the sort of big picture of where you're going. And that's another thing you really want to consider is that the likely thing when you're giving a chalk talk is that there's going to be maybe 1 or 2 people who are like pretty familiar with your area, but most of the people in that room are going to be, way less related or not going to be working on a similar area. So you want to make sure that that talk is quite accessible. So that was really great. Thank you to Johan and Rui for presenting their very quick ones. I hope that did either of you know that this was coming? Because I did this a couple of years ago. Hopefully not. So that was even more impressive that you were already prepared. Okay, so I'm going to start sharing my screen again to go over a few more things, and then I'm going to open this up for more questions from the audience. So. See, where were we? We did the content. Can you see my slides again? Laura, you can see? Okay, great. Okay, so I like to put this on some Chalk Talk do's and don'ts. We kind of went over sort of the major, what a Chalk Talk is. So the first thing to remember is that you relax and that you're the expert in the room. Okay, so one of the things that happens is people tend to go into these things and feel a bit afraid or intimidated by those who are in the audience. But the truth is that you know your subject area more than anybody else does. So you should be confident going in and not try your best to not be intimidated by the audience. The best way to do that is to be prepared. So we talked about it. So practice your seminars, practice your Chalk Talk, ask your colleagues to sit in for a Chalk Talk session. The other thing is that it will come up that you'll get some tough questions to ask. And I think the important thing to remember is that it's important that you can be deferential to your speakers, so you can, or to the people who ask the question. So, but you should also be firm when you have an answer to their question that you may even disagree with them. Because in the end, it's more important that you not agree, but that you show that you can that you can defend your positions and that you can defend that, no, my approach is the right approach. So they're not looking for somebody who's going to be a pushover, right? They want somebody who's going to be a leader in their field. So it's important that you're able to demonstrate that your ideas are good and that you really stand by them and support them. Another thing you have to keep your eye on is that you have to really control the pace of your seminar. What do I mean by that? Well, you may start getting questions within the first couple of minutes in your talk before you've been able to even talk about the major aims. So you want to make sure that you have a way to stay on track. So if you think you're going to start to get sidetracked by some questions that are taking you far away from what you want to talk about, it's okay to address them and to say things like, actually, I may have to answer that question later. I want to stick to the major things I want to talk about first. So it's important that you remember to control the pace and that you control the direction of the talk. So don't allow yourself to get too sidetracked by questioners. Okay. So you have to practice. How do you answer a question that may be sort of peripheral, but that you don't spend too much time so that you can really control the direction of where your talk is going. All right. On that line, one of the things you don't want to do is to not get flustered by really tough questions. And you have to remember, or by tough questions or by tough questioners, because you will find that some people in the audience can seem combative. And I've had this happen where it's the same person I go to at every conference, asks the same question, and I know he's trying to get under my skin. You have to remember that it's not personal, even if you think it's personal. And you just have to learn to answer those questions. And again, move on and control the pace of the talk. Okay. This will happen to you at some point in your career, and you don't want to rise to the bait. So I'm sure most of you have been at a symposium where there's been some discomfort. So if you feel that happening in your seminar, just answer nicely, and then try your best to just stay on track on what you want to talk about. The second thing is that it's okay to say you don't know. I've had it happen before when questions happen in a symposium, and you can tell that the person who's answering, or that the person who's giving the chalk talk, is not being honest in their answer. So it's perfectly fine to not have an answer for a question. An even better thing to say is that it's not known, like nobody knows. And I have to be careful saying that. You have to be pretty confident, but you can say, actually, there's nothing in the published literature, for example, that would support that. So I think it's important to be able to say, when I don't know, that's fine. You can say you don't know, or you can say it's not known if you're comfortable in the literature. And then the other key thing you don't want to do is to let the meeting get out of your control. This goes back to something I said earlier about controlling the pace and the direction. I've seen some of these chalk talks go where the person never gets through their first aim. And it's because they let the audience drive the discussion as opposed to controlling it themselves. And in most cases, this is not because an audience member is trying to derail you, but it's because if you get 20 scientists into a room together, chaos can ensue, right? Everybody starts asking their own pet questions, and it can be very easy to make it so an hour will be gone and you barely talked about like one of your aims. So you have to remember that you're really in control of the chalk talk. Okay. The other thing that you want to consider that you might want to highlight is that, and this all goes to the first slide when I talked about one of the goals is to convince the audience that you're ready to lead a lab, is that you may want to mix in that some of the things that you may have already done, which is like mentioning that you've already established collaborations, especially in the case of when these collaborations are distinct from your postdoc mentor. Because typically what's going to happen during these during the seminars, they're going to try to address the question of whether or not you are independent from your former boss or from your current PI. And by showing that you've established independent collaborations, that's one way to do that. The second is by just flatly mentioning that you've had discussions with your PI, we've discussed which areas we're going to pursue, and that we're not going to compete. So if you have a concern about that before you go into your chalk talk, I highly recommend you having that discussion with your PI so that you know with certainty that you're not going to be working on similar areas. So at this point, I really wanted to that's really all of the material that I had to present. I'm happy to answer other questions that you may have about the talk itself. So I'm going to stop sharing. It would be great to see some faces, see people ask some questions.
Hi.
Yeah.
So I have a question, like how much in detail we have to go in methodology?
I would say as little as you think will get the understanding of what you're doing. But so this is going to be very field specific, so I don't even want to give examples, but you want to say like in my example would be like, I'm going to knock out this particular gene and I'm going to measure this particular thing. Like I don't have to describe how I'm going to knock out the gene, like, how I'm going to do the measurement. If it's something that's pretty common, like if they want more details, then you let them ask you for more details.
Okay.
Does that make sense? So you want to be able to give the minimum details that you think most of the people in the audience will be satisfied, that you can achieve that, because they really want to know that what you're proposing is feasible, right? So if you're talking about a commonly used thing, I'm going to do ChIP-seq on this. I'm going to make a mouse knockout. Like these are, most people understand that that's pretty trivial these days. So when it comes to a particular assay that only your lab in the world does, you may actually want to spend time walking through that assay, right? Because in that case, that's also what makes you unique. So you want to highlight that. So it really depends upon the individual.
And like if you prepare a like presentation, let's say like chalk, not the chalk talk, like the whiteboard or the
Yeah.
blackboard. So how will you go into like, how you differentiate your seminar talk? I know seminar talk, you are just going to say like what you published, right? But the proposal talk is how do you need to give that much, basic information if your field
Ah,
is like totally new compared to
yes.
the seminar versus the, or
Right.
you can,
Yeah, thanks for asking that question. It's something I forgot to actually mention. So in the most cases, the chalk talk is on a separate day. Not always, but it's usually on a separate day, and
Hmm.
almost certainly is going to have different audience members, right? Just
Mhm.
because of people's schedules. So it's very important at the start of your chalk talk that you also highlight enough background so that they get the gist of what you've done before. So that's actually mixed into the introduction when you're describing before you get into all the new aims, you definitely have to lay out a little bit of what you did in the seminar and like your main seminar.
Okay.
And that amount is probably 10 minutes, I would say. Sometimes it may be a little bit more because you may need to give more background information that describes the rationale behind the aims. Okay,
Yeah.
so
Mhm.
that is actually something that's quite important to do. So you have to make sure you build up enough time in the chalk talk. If your chalk talk is like immediately after your seminar, that may be the one case where you don't have to do that.
Yeah, yeah.
Yeah.
That makes sense. Yeah.
And usually at the end of your seminar, you're going to do a very quick slide that talks about your future aims. So because there may be some people who will not get to see your chalk talk, but you want to
Mhm.
make sure they understand where you're going. So
Mhm.
likewise, when you give your main seminar, make sure you have a slide or two at the end that
Mhm.
teases what your direction is.
Okay.
Yeah.
Sounds
So
good.
that way you kind of have your bases covered in case some people in the audience can only go to one or the other.
Yeah.
So good question. Thanks for asking
Yeah.
that.
Thank you. Yeah.
So Tasha, you have a question?
Hi.
Hi,
So I
Tasha.
had a question about the presenting or discussing the timeline, your short-term versus long-term goals. Is it important to actually draw that out during the chalk talk?
So you could do that. I mean, so I've seen that done. And, when you write a grant, a lot of times now, you kind of putting timelines into like your grant proposal.
Yeah.
And because you've, presumably have sent this in with your application materials, at least the committee will have access to that. I've seen it done and it's absolutely fine to do it. It's fine if you want to leave it out. I think, it's reasonable at one point to say like aim one, even if you don't draw it out, to say this is like in the first year or two, this is stuff we've already got cooking. So it's going to be the first thing that we tackle. And then, aim two is going to follow shortly behind, or it's going to be along with it. And aim three might be slightly downstream. And at some point you might even say like, but in 10 years, this is where I want to be too. So you can also add something that's not in your grant, but like, where are you going in the long term? So that's good also. So whether you want to draw that out on the board, I think that's up to you. I think you're going to kind of it's good to sort of hit those points in your proposal, but then again, in your talk.
Thank you.
Or you might get asked that specifically. So someone in the audience could just ask like, how long is this do you think this is going to take? Or yeah.
Okay. Thanks.
I can't tell who asked questions first, so I'm only going to go from left to right on my board. Let's see. Akemi, is that right?
I think there were two people before me. I
Oh,
was the
okay.
last two. I'm gonna go after them.
Oh, okay,
Yes.
sure.
I can
Johan?
actually see it, so I'm happy to kind
okay.
of jump
Yeah,
in.
go ahead.
Okay, Virginia, go ahead.
Hi, how are you? I have a question that may not apply for NIH institution, but can you comment on how and where add information about when are you planning to submit your first grant? How many people are you considering to put in those grants or things like that? Mainly for those institutions that they are asking you to bring, I don't know, K99 or things like that.
Sure. So that's really tricky right now for NIH. As you guys all know, there's like major upheaval in how extramural grants are going. I'm trying to follow the news, but I can't keep up with it. And I'm not applying for NIH grants. So I think one thing you might want to do is to reach out to some mentors who are in the extramural community. So presumably you all went to grad school at some point and have a former boss who's working, or people you can reach out to in the field. Yeah, I would recommend emailing them and asking about what are they doing with grants. Talk to people who are in extramural. And if you're applying in Europe, that's even easier. You can look at things like ERC, when are the grant deadlines? Again, talk to people at those institutes that, particularly junior investigators. There's nothing wrong with saying, like, I really am interested in, these institutes. Find junior people who are there, ask them, hey, how did you get your first grant? Where did you apply? So in most cases, it's like word of mouth or going to the major funding agencies' websites. But those can be difficult to navigate. So I think talking to real people who are like really doing the applying, that's probably the best place to look. Yeah. I mean, NIH is changing every day. NIH is changing every day. That's the problem. Yeah.
We have one in the chat asking about how much or how little should you talk about your aims during the seminar, and do you worry about overlap?
Yeah, I think a little bit. So in your seminar, you want to talk about your aims very little, only at the very high level. So not a lot of details, maybe just the big questions. So I would say like 5 minutes to 10 minutes at the end of your seminar. Yeah.
Great. Well, I said I can see, but now I can't see your name. I'm sorry. Johannes, go ahead. Oh, we're not hearing you.
Someone's muted.
I see that you're unmuted. I'll let you.
Can you hear me now?
Yeah.
Oh, yeah,
yes.
that's.
Okay, got 3 different buttons to get unmuted. I just wanted to pick up on 2 points which you made earlier. One is the format of the Chalk Talk. It sounds like that the format depends a lot on the institution. Well, rather seminars usually have a particular format. How different can this be? I mean, in your experience, I mean, when I think Chalk Talk, I think about drawing on a board, but you alluded to the fact that's not always the case. So what to expect there is, I think, a question. I mean, can you go with one format, or do you have to adapt every time they invite you to a Chalk Talk?
So I think you should be ready for multiple formats, especially if you don't ask ahead of time. This is why I just recommend, like, what's the room I'm going to be in? Is it going to be a big or small chalkboard? I mean, sometimes, the room kind of defines like how big you can write and how far away people are. I mean, this is a simple thing, but if you don't think about this, your text may be way too small for anyone to read, right? So like, and some of the NIH spaces that we've used have been really horrible for Chalk Talks because the audience is too far away and nobody can read what's being written. So that's why I say, like, you be ready to write big, but you might be in a small room with a giant chalkboard, which is great because then you can write big and everybody can see it. I've seen ones where they actually want a PowerPoint. So, you could actually have a PowerPoint done for your Chalk Talk. So most of mine, I did 5 Chalk Talks when I was on the job market. I think 2 of them were whiteboard, 2 of them were like chalkboard, and 1 of them, I think I was allowed to have a PowerPoint. So there you go. I had, 3 different types out of 5. But I would say, NIH, all the ones that I've done as Statman have all been like whiteboard style, but the rooms have been very different. So just something to remember. I mean, just to think about. So, yeah.
The other question was about the handout that you recommended. I mean, should the handout essentially be like my cheat sheet where I have my schematic on and have the text appropriately where it goes, or is that?
Yeah,
what's
I
the
love that. I
recommendation?
mean, I think that's a great idea. It could be more or less detailed. I just like, again, some people will not bring they're just going to show up with nothing. Maybe some of them didn't even read your proposal. So it's nice. I have always found it's kind of nice that they have something else to follow along. In case they can't read the board that you're writing, they have something that they maybe can see. So I like that idea of almost doing like a version of what you're drawing, but it could be any number of things, I think. And just keep it simple. I mean, you don't want to have a 5-page book there. You want it to be like a 1-page something, so they can see it. Yeah.
Right. Thank you. Thank you so
You
much.
don't have to do this. I mean, you don't have to. I've had people do it and I like it, and I did it myself, and I think people appreciated it. So that's based on my, limited experience, but I enjoy when someone hands me something, and I also did one of these when I get mine.
Thank you.
Yeah.
Renee had a good follow-up to that in the chat, which is, does the advice that you've given and what you talked about change if you are giving a PowerPoint instead of drawing on the board? Like what changes in that scenario?
Yeah, I mean, I still think, you're not going to have as many slides as you had for like a seminar because it really is going to be more of a discussion than a talk, right? So you're going to have a lot more questions. So if you're going to do a PowerPoint version of a chalk talk, you don't want to have a huge number of slides. It should be like 5 to 10, whereas most of you will probably have 30 slides plus for like an hour seminar. So that would be my main suggestion, is just it's not a huge amount of material.
Thanks. Akemi, go ahead.
Thank you, Todd. So I'm an epidemiologist. I have a very broad research program spanning from screening, diagnosis to survival of the whole cancer care continuum. Now, presenting that, and there's I'm working on endometrial cancer. There's not a lot known about the field. So my mentor right now at NCI, we're primarily working on the first phase of the cancer care continuum, looking at diagnostic biomarkers, that aspect of it, screening. And my work primarily focuses on, and what I'm trying to apply for the K99 and RO, the latter part, the survival part. Now, when I'm presenting to a faculty position, I would like to present the whole thing, but this is broad. Would you suggest picking one over the other, or present in the whole spectrum of different things that I'm working on? And then the other challenge is that most of my work is kind of stuck with one collaborator, which they have the largest sample data, that I can't necessarily have that depth of data from other different available sources. There's not a lot of diverse populations in other data sources. So stuck with, in terms of data accessing, I'm stuck with one main collaborator. So there are a couple of challenges, and how would I kind of narrow it down when I'm applying for a different institute other than the collaborators?
Yeah. So, I mean, so assuming you'll still have access to that collaborator, right? So I would sell
Yes.
that. Yeah, I would sell that as a strength, right? There's not a lot of people who have access to this. I have access to this big data, and this is an important collab. Like, you have to be honest about what you're going to need to be successful. So in that case, I would sell that as, this is a real strength of mine. I've established this really great collaboration that has a huge data set that's important for my work moving forward. And I think that you just sell it that way. Now, with regards to the depth versus breadth problem, I think everybody here understands that problem, right? What do I mean by that? How much breadth is in your research program? So how wide of a research area are you studying versus how deeply are you studying one thing, right? And everybody has to draw the line somewhere, okay? It's hard for me to say exactly where you should go, but it's like when I see people who are too broad, I know it. And when I see people who are too focused, I know it, right? So the answer is somewhere in the middle, right? You want to be sure that you're not too broad or you're not too focused, or else you'll get somebody who dings you on a grant application. This is true of all the grants you'll ever do. Someone, more often than not, it's too broad. I would say that tends to be the bigger problem, is too broad. But I think what you want to do in your Chalk Talk and Symposium, you're focusing on your one grant, which is presumably going to be sort of focused, right? It's 3 aims or 2 aims, but you want to, in your long-term goals, think about like where you could broaden into, right? So you want to make sure that you can tell them that, look, I am really focused for the short term because that's how I'm going to get tenure. It's like, is my R01 aim going to be a success? Maybe a second R01 is going to come along, and that's really going to be what gets me tenure. But if I'm going to be a great colleague, I have to be able to describe like, how can I synergize with the department? Where are my long-term directions? How am I going to impact clinical medicine? So you have to be able to sort of navigate both of those. You have to say, look, I'm focused enough, but I have broad interests too, and I'm going to impact a field. So, you have to do both.
Thank you so much. So
Yeah.
follow up on that is, so in my K99 training portion of it, I'm focusing on the latter part, survivals, very specific exposure and outcome associations. In that, I'm saying that I'm also working on with my PI on the discovery of diagnostic biomarkers, and that's primarily his research program. I'm collaborating and getting the expertise that I apply for the latter part of it, where that is distinct from my PI. That's how I'm pitching it.
Yes, I think that makes perfect sense. Yeah. So, I mean, and just be aware of most institutions, like I can tell you, I'm also on the central tenure committee here at NIACHD, or I'm sorry, at NIH. So, you know, they really want to see papers where your previous mentor is not on them. So that's, you know, they just count less, you know what I mean, towards your tenure in most cases. Now, there are rare cases, and I don't want to go into all the field specific things. There are some fields where it's impossible for that to happen. And those are taken on a case-by-case basis. But I want to keep this seminar broad, so most of you should be thinking about how am I going to disentangle myself from my previous mentor? Because, you know, you're going to have to move to papers where you're the last author and your former mentor is not on the paper. Like I said, there are case-by-case things, so I can't go into everybody's individual thing, but that's generally what's going to be appropriate for most of the audience.
I've learned different about having your PI or mentor on papers or something. So NIH, it's different. I feel like all that I've learned from extramural, they would suggest that the most senior PI should be on the papers to show that on your grants that you're applying.
Yeah, well, you should be the
different from
senior author.
NIH.
Yeah, so let me rephrase that. You should be the senior author on all of your papers, last author. Okay, yeah, and your previous PI should not be on those papers as much as you can. That's the tricky part for some fields. But the point is that that's what's establishing your independence. So you'll get a question by someone in the audience. Have you talked with your previous boss about this? Are you going to work on the same problem? Who's going to get credit? Who's going to write the paper? So that will be something that comes up. So you have to have those discussions with that PI, and you have to be ready to answer that question. And I can say on the Central Tenure Committee at NIH, if we see cases where most of the papers that you're listing as your top 5 papers have another NIH investigator that's listed last, that's going to be questioned. So you want to avoid that.
All right. Thank you, Todd. Yu Ying.
Hi, I actually put it in the Q&A, but it probably didn't go through. So my question is, you mentioned that 3 aims are like 3 aims is a bit in your R01 grant. And I'm wondering how you like the idea of instead of like within tied to 1 grant to have it as like a short-term, midterm and long-term goal of how you want to run your lab, like say in the next 10 years, or even 3 different areas that you want to work on so that you can let people know like at once what are the different projects or questions that you want to tackle that don't necessarily relate or fall into 1 grant.
Right. I think that's a reasonable way to approach it. I think that if you're going to do that, you have to be careful, though, that you're not looking like you're too broad. Because if you're going to tackle 3 kind of very different areas, most people in the audience will say that that's not realistic for a small lab. So when you're just starting out, right, you're going to have a small lab just by the nature of hiring people. Like even if they give you a ton of resources, it's going to take 3 years before you fill 3-4 people into your group, right? That's just the nature of how you can hire students. So I think that's good if you're going to say these are the 3 big areas I'm interested in long term, but then whittle it down and say, but here's going to be the focus of my lab during my tenure track period. So that they're going to say that, look, that's a very feasible set of questions that you can address with a lab of that size. Because if you try to say that I'm really interested in these 3 big things, but I'm only going to have a lab of 4 or 5 people, there's no way you're going to be able to address all those questions. So that's going to sound like you don't know what you're talking about, right? Because they remember, you're trying to convince them that you're able to achieve your goals. So they have to match if they're long term, if that's a career goal, like in 30 years, I want to have answered these 3 things. I think it's okay to start that way, right? There's a little bit of a danger to it though, because science also changes when you're talking about those timescales. So I usually say like, you have to find that, this kind of goes back to my previous point, you have to find that balance between how broad are you going to be and how focused can you be when you're actually starting your lab, because that's mostly what they're going to be looking at is are you able to get started and get papers out right away? I didn't really answer your question. Everybody has to answer this question for themselves, right? But you have to be careful, as that's my concern.
We have one in the chat
Yeah.
kind of asking about what you talked about in terms of controlling the room and controlling the pace.
Yeah.
Does it make sense to set out a timeline at the start of your talk? And so, for instance, this person is saying, should I say, I'm going to give an outline of my R01 in the first 15 minutes, and then I'm going to take questions?
I think that would be a lovely way to do it. I haven't seen anyone do it exactly that way. I think it could work. I mean, most people in the audience are up for having some structure. But you may get some people who just derail the structure. So I think it's reasonable to say, like, here's my goal, right? My goal is to like give you a 10-minute thing, and hopefully, we can focus on the questions as I get to this part. I think that's very a reasonable approach. I like it. It might not work. It might not work.
See what your mileage is. Yeah.
Yeah, that's right.
Okay.
Yeah.
Thanks. Johannes.
Hi, can you hear me?
Yes.
Hi, sorry, it's me again. I wanted to pick up on something you talked about in terms of, separating yourself from your former mentors.
Yeah.
I mean, particularly in the beginning when you're small and don't have that much money in the pocket. I mean, in a field like mine, having these collaborations with former mentors means having access to specialized equipment that is not necessarily available anywhere else.
Yeah.
In my case, we're talking about a phenotype or cage equipment that costs $250,000 to buy new.
Yeah.
So these are things which I want to have immediately available. So on that basis, is it a justifiable rationale to maintain these collaborations for papers with the look on tenure? Because it will be some time before I have the money to have something like that on my own.
Yes, absolutely. And I said, particularly if those collaborations are not your PI, right? So I didn't want to oversell that point, that you have to distinguish yourself from your PI. But I also think it's very important to say that you've established these collaborations that are going to give you a leg up on the competition, right? So that's a great case to say that, like, yeah, you want to celebrate your collaborations that you've established. It says that you're ready to jump out into the field. You have a support network, right? That's basically telling the people in the audience that you've already have people supporting your work. So I think that's a great use. I would spin it that way.
All right, thank you.
Yeah, absolutely. Yes. And for those of you who are early in your training, in your postdoc training, this is actually a real important thing, is that it's important to think about establishing those collaborations independent of your PI as you're going through your postdoc. Now, as a mentor myself, I encourage my people to make connections with others, and I always let people do collaborative stuff that's independent of me. Some of your PIs might not do that, but it's certainly very reasonable to think about establishing those kinds of collaborations as you move through your postdoc. Normally it happens kind of naturally because labs, who work near each other tend to collaborate, and NIH is a very collaborative place, but it's very nice if you can highlight those collaborations that will basically follow you as you go on to other places.
That's everything that I see. Does anybody have other questions that I either missed in the chat or that you didn't quite formulate yet?
I have a quick question.
Oh, yeah, go ahead.
Sorry, my camera's not working, so I'll just have to be a black box for you. I'm just trying to get a sense of understanding what the role of the chalk talk is, in the sense that you gave the impression that some people showing up might not have read anything, in regards to the aims of the application. So in that sense, it feels like I need to educate those folks about the entirety and the detail. Meanwhile, others may come in, maybe be even familiar with the field, and there I might have the instinct of wanting to pursue, higher minded ideas or future oriented ideas. So how do you balance those two different audience members?
Yeah. So that's the challenge of every seminar you'll ever give for the rest of your life, right? I always say that this is like the challenge, and it doesn't matter what kind of talk, it's to always know your audience, right? So that's why it's also reasonable to ask who's going to be in an audience. I mean, sometimes the chalk talk is really limited to just like PIs in that exact department. Sometimes they let postdocs in. I mean, so at NIH, we've done some of these where we let our own postdocs sit in, but we don't but like they're maybe not allowed to ask questions. So like it's kind of important to know who might show up. But I always think that I've never heard somebody who said that the talk was too easy to understand. Have you ever gone to a seminar that you thought was too easy to understand? No, it's always that was too difficult to understand, or I didn't. So I always say accessibility first, right? If you have to err on being either too detailed or more accessible, always err on the side of being more accessible. I think that's a key lesson for any talk you give. And then naturally, people who want more details will hit you up for those details, and that's a very appropriate time to get more detailed. So in that talk, does that make sense? In that chalk talk, you start very broad, start very big picture, and then move to more details, and the limited details until they need more, until they start, pinning you down to ask for more details.
Thank you.
Yes.
And we had a couple of questions about the slides. So Todd, I'll ask you for the slides and then we can get those sent out.
Oh yeah, sure. Yeah, absolutely. Yeah. And unfortunately, so I'd love to be able to say, oh, everybody send me their Chalk Talk ideas. Unfortunately, there's too many of you and I can't look at everyone's individually. But if you guys have specific questions that can be answered in a reasonable email, I have really enjoyed follow-up to these sessions. If you have suggestions for me, you have questions that you didn't want to ask in public, but you might just want to ask over email, I am really happy to answer those questions. So don't be afraid to reach out to me, and reach out to people in your own departments too, because most people at NIH have served on committees. They can tell you about good and bad Chalk Talks. And I think it's reasonable to once you've been through a couple of these, you kind of have a feel for their purpose and which ones are good and which ones aren't. So I hope I've been able to at least make it to demystify it a little bit. Really, it's your chance to have fun. And the way I sort of approached this when I went into mine was, when am I going to have the chance to have 10 really good scientists like really grill me on my work? So take it as like a learning experience. This is how I'm going to improve my communication skills, the way I think about grants. So I only ended up getting, like, I think I told you guys, I went to 5 Chalk Talks. I get 5 seminars. I got 2 job offers and a couple of second places. Okay. But I've maintained friendships with many of those people that I didn't get jobs at. So those are also great experiences for you to just get to meet people. And I can tell you that 3 of those people at places I didn't get jobs all wrote me letters for my tenure package. So, it's not always just about getting the job. I know it feels that way because there's a lot of pressure, but it's also about just networking. These are great networking opportunities. So don't waste them. Be prepared, get to meet those people, make a good impression, because even if you don't get the job, you may actually find people who will be beneficial for you in your careers. Maybe that's a good way to end it. So
I think that's perfect. Todd, thank you so much.
You're welcome. It was a pleasure.
And thank you, everybody. We wish you very good luck. Do reach out if you have questions, if you want feedback. You can always reach out to OITE as well. So thank you all.
So long, everybody. Good luck.